session, we have mostly talked about the MaxQuant data analysis, right? That you have got some raw files or you have generated some raw files and you want to know that what are the proteins that got identified in that particular files, right? So in that case, as I have discussed, that you need the UniProt database file. For example, if you have the raw files from the human samples, then you need to go for a human database file, and then you need some parameter file and obviously the raw files that you want to generate. So now let's assume that you have completed your MaxQuant analysis, and I have showed, right, like how the MaxQuant output file look, and it's contains a number of columns. So once you have clean up that particular column, that means that particular messy you have cleaned up the messy data, and finally you have got the P cross N matrix, right, where you have the protein names in column A, and the next all the LFQ intensities in the next columns. Then we are ready to go ahead with the next part of the analysis, which is the secondary analysis part after the MaxQuant analysis, right? So for this particular statistical analysis, you can use any software that you want. For example, you can do all these steps of the statistical analysis in Python. You can do all this analysis in the R, and there are other softwares are also available which you can use. But I'm showing the hands-on session in Metaboanalyst. The reason is that this is a very compact software with almost all the the parameters, statistical analysis parameters present, and also this particular software is a user-friendly online software. So I will suggest you guys to keep keep all the files that we have shared with you ready for the analysis. And first, I will ask you to open the file 3, that is the Metaboanalyst input file. And before going into the Metaboanalyst directly, I will try to spend some time with regarding the input file. So we should be very much familiar with the file that we are using for the Metaboanalyst or any kind of statistical data analysis. So in this particular file, as you can see in the headers or the columns, all the samples names are there, right? There's sample 1, sample 2, sample 3. That is the sample unique names are there. Whereas in the second row, you can see the groups are there. That means this particular sample belongs to which group. And in the column A, you can see the gene names are there. So as you have seen in the MaxQuant output file, there is a column A was having the UniProt IDs, right? So you can take those UniProt IDs and you can go to the UniProt and convert those IDs into gene name, and you can place those gene name over here, and you can do the analysis. The reason behind choosing the gene name is that when you will be make some box plot or heat map in the particular Metaboanalyst, the gene names conveys information more rather than UniProt ID, because gene names are very much familiar to us, right? So this particular file format is a P cross N matrix where the features are there in the in the rows, and in the headers or in the columns the samples are there. So this particular format is very important for putting as an input file in the Metaboanalyst. And that is the reason I show you the output file of the MaxQuant, which was having so many columns, and actually the next input file of the Metaboanalyst. That means how you need to convert that particular file into this particular file before doing the next part of the analysis in Metaboanalyst, right? So this particular input file, what I was showing in the P cross N matrix, is not only for Metaboanalyst. In most of the data analysis platform, and also in Python and R, this kind of formats and this kind of file formats are very much important and very common, right? So I think you have already searched Metaboanalyst in the Google, and once you are clicking that particular Metaboanalyst, you are seeing this web page. And I will request you to click here to start. And once you are clicking here, then you will get the next page, which looks like this, right? So you can see that how many different kind of options Metaboanalyst give you to do different dynamic analysis with your omics data, right? And you must know that this particular software is not only restrict to proteomics data, rather it is very much used for metabolomics data. But you can, yes, you can use this particular software for accept some of the parameter for proteomics data too, right? So today we will try to explore the statistical analysis part. So I will request you all to click into this particular statistical analysis part. And once you are clicking this particular statistical analysis part, you will get the upload your data page like this, right? I hope everyone has reached to this particular page. And once you have reached this particular page, I will request you to choose the files, the same files that we have shared with you, right? So once you have clicked into the file, and make sure that you are use this particular drop down menu and you are using samples in column and unpaired. So what this options tells you is the samples in columns means all the sample informations are in the headers or columns of the files, as I have showed you in the input file, that all the informations of the sample names and the groups are in the header. And it's unpaired obviously because all the samples whatever we have are all these samples are from different patients. So these are unpaired samples, right? So once you are putting the choosing this particular option and choosing the file, you will see that the particular file name is showing over here. And make sure that you are choosing the correct option from the drop down menu. Otherwise, you will find an error. Once you are done this, please click submit. Once you are submitting this, the next part will be kind of a data integrity check page that Metaboanalyst gives you, and I will try to go through each line, whatever the data processing informations are available. So, for example, the first is the check data content passed. That means you don't have any error or you don't have any messy data kind of thing in your particular file format. Then the samples are in columns and features in row. As I told, the genes are the features over here, and they are present in the rows in the column A. And the samples are present in the columns, right? The uploaded file format is .csv. That is this is also important, part, that the file format which we have shared in .csv format, and Metaboanalyst only take .txt and .csv format, right? So the next part is that the uploaded data file contains 20 samples, and and it is having two groups and samples are not paired, right? So now if you go down, you will find other informations also regarding missing values, because I have whatever the files I have shared with you guys are having no missing values. But in proteomics, generally, you will not find generated data without missing value. So missing value is a very common thing, and in the next slide I will talk more about the missing value part. So let me give you a brief about the missing values. So whenever like you will analyze some data in the MaxQuant, and whoever has done proteomics, they have find that missing value is something which is very important part of the like very common part of the proteomics, where you will find zeros or blank data in most of the in many samples of a particular features, right? So how these missing values are coming? So missing values are not like biological, because it's not like that the particular protein is not actually expressing in that particular patient or that particular sample. It's something that the intensity of that particular protein is not that much that it is crossing the noise is to intensity ratio, right? So this is a very important thing, that in the mass spec or in the MS analyzer, we need to put a threshold. That threshold is an intensity threshold that don't allow a particular intensity, if it is less than that, to present as a intensity. So this is something which detected as noise, right? So this is a very important part. And then what to do with this missing values, because it is very difficult to do any statistical analysis with this with the presence of this missing value, right? And I will request that you guys should go and read these two books that are available online, and this will give you many information regarding the missing value imputation, types of missing value imputation, and how to what is the best way to estimate these missing values. But in general, I will try to give you an idea that missing values there are three different types of missing values. First is MCAR, that means the missing completely at random. Next is MAR, that is missing at random. So this particular two thing, MCAR and MAR, are the missing values which are kind of, which we are not much bother with, because this particular missing value can be, can be solved with different missing value imputation techniques and estimation techniques. But the one which is not ignorable and is really very much we should worry about is that MNAR. So this is missing not at random. So this is something, this particular missing not at randoms are something which where the missing values are for a particular sample is a lot of value for a particular sample is missing, whereas out of, so for an example, I have given you 20 samples, you are finding one sample which is having 40 or 30% of the missing value, whereas the other samples are not having this particular missing value. So this particular sample is having this missing value, and it surely make a problem in the next part of the analysis, and the missing value imputation will really not work for this particular samples. And why this is happening, this can happen due to many reasons. Like for example, in the sample processing, this particular sample has not been processed well, or while bringing the transporting the sample from from your hospital to your lab, it might happen that that particular sample gone bad or has not been stored properly. So this could be a reason, that is the reason this why this particular missing particular sample is having so many missing values. So as I discussed, that MNA or something, or missing not at random, is something which should not be which should be taken very seriously, and should not be ignored, right? So let's come back to the Metaboanalyst software. And when you are clicking into the missing value estimation, you are getting a link something like this, right? A page something like this, right? So the first step is that remove features with too many missing values. That means when there is too many missing values, how many missing values you want to take as a threshold, right? For example, in many publication, I have seen that people take 30%, only take 30% missing values, right? So whatever 30% missing values are there, that will be in, that particular missing value cells will be filled with the data from the other 70% of the data. So this is a strategy people use, but I will suggest that while screening your data for the first time, go ahead with a 50% missing value and try to see that how much features you are losing, how much features, how more features is having missing value. So this is an important steps you should take into account. And the next part is that estimate the remaining missing value. So for example, if you are having 50% missing value, or you are having 30% missing value, what strategy you will use to fill this particular missing value? So this is in proteomics, mostly people use median imputation or KNN imputation, though there are many other imputation strategies are also there, but KNN imputation is something which is widely used in proteomic data analysis. Now I will request you to go to the come out of the missing value imputation estimation part, because we don't have missing values, and click proceed, and skip this data filtering part, and just put it as a none and proceed with it. So the next part is having the sample normalization and sample transformation. So this is again an important parameter in proteomics data analysis, even in metabolomics data analysis. But you should understand that why normalization is very important. So you are running samples, one after another in your mass spec, and though you are trying to keep all the parameters, all the sample processing part very much, reproduce very much same, but still the samples might have, the variation in the central tendencies, right? The mean of this particular samples might deviate from one another, and this deviation might lead to, a bias while you are choosing the best proteins or the differentially regulated protein at the end of the analysis. So it is a better it is a better suggestion that you go for a normalization. And there are many normalization strategies are available, but I will request, as you are starting for the first time with the analysis, I will request you that you start with the median normalization, which is a very common normalization strategy. And the next part is the transformation. As you have seen the the data in the input file, that these are present in e⁸ and e⁸ The intensities are pretty big, and it is really very difficult to to handle these big intensities, and it is very difficult to plot any distribution curve with this particular intensities, right? So it is better to transform your data, which will help you to plot a distribution curve, and it will help you to understand whether your data is skewed or your data is normally distributed, right? So once you are doing this and clicking normalization or normalize, you will find these two options, sample view and feature view, right? So in this particular thing, I will request you to go check the sample view, because feature view is something which we are not that much bothered in particular this kind of analysis. Rather, sample view is important because we want to see whether all the samples we are taking forward are central are normalized or not. So as you can see that after normalization, all the central tendencies are normalized, and if you scroll down, you will find a distribution curve which is giving an idea that our data has almost normally distributed, not completely, but yes, there it is normally distributed, and the data is ready to take forward, right? So let's now click proceed. So before proceeding, I will just want to tell that I got many questions, and I get many queries regarding normalization and batch effect, right? So I must say that don't confuse these two things, that the batch effect and normalization. Normalization is different and batch effect is different. So to make you a little comfortable with the term batch effect, I'm taking a reference figure from a manuscript, and I will try to show you that how batch effect actually happens and what is the meaning of this. So for example, you are working in a project where you have 200 samples or 250 samples, right? And you are not getting these samples at a go. You are getting these samples in batches, and you are processing these samples, running these samples, and generating this data, and you are keeping this data with you. Again, when you are getting samples, you are doing the same, right? And while doing this part, it might happen in the sixth day or the sixth batch or the seventh batch, your instrument breakdowns, and you have again fixed the instrument, or you have changed the columns. And while you have done this, you find a different batch, different batch effect, kind of different batch, different variation in those particular samples, as you can see over here, right? So this is something which we call batch effect, and it is really very important thing to look into your data when you are having a large amount of samples and you are running those samples in different batches. Especially in Metaboanalyst, people do that they run samples in batches, and they encounter this particular batch effect problem very much. But in proteomics also, we find this problem very common, and we should not ignore this problem, because once this batch effect is there, it might happen that while doing the statistical analysis, and finally when you are getting the proteins, that is a differentially expressed protein, this particular batch effect may work as a culprit, and you are getting the proteins in terms of some technical biasness. So in one of the, I want to show you some live example. In one of the project, I was doing some metadata analysis where I took some data from the repository, and I tried to see, though these particular data sets are from same diseases, the grades are also same, but still you can see that there are so many the studies are so different from each other, right? And this is something which we call as the problem of batch effect. Though these are meta-analysis study where the studies has been run in different labs, the different instrument, but still this is a big problem. And in this kind of data, normalizing the data or even batch correction data don't work. So that is the thing I must say, that please look into your data or whatever data you are downloading, that whether your data is having any batch effect or not. So if you have encounter batch effect, Metaboanalyst don't allow any batch correction, but yes, softwares are available, like for example Combat, which works on the principle of the Bayesian statistics, and it helps you to do adjust the batch effect, right? So I will request that I have already shared some material regarding the Combat and batch correction. I will request you guys to go through it and to understand the batch correction and batch effect more genuinely. And you must know that this particular batch effect happens due to technical artifacts, right? So let's come back to the Metaboanalyst and let's come back to the normalization window. And as you can see that our data is normalized, and I will request you to proceed. So once you are proceeding this thing, you will get a multiple options of different visualization plots and analysis that Metaboanalyst gives. And I will request you first to click into the PCA, that is the principal component analysis, right? So this particular, once you are clicking this PCA, you will encounter a window like this, which will have PC1, PC2, PC3, and some percentages are there, right? So this is a very important part of the analysis, and I must say that PCA is something which is a very powerful clustering model, and it will give you information about your data, and it will help you to interpret your data to a large extent. Even if there is a sample which working as an outlier, PCA can tell you that this particular sample is an outlier or is having some difference when we are comparing with other samples, right? But in one hand, we must know that one PCA is a clustering techniques, and so this is a unsupervised model, right? So the unsupervised model is something which don't take the groups into account. So if you go back and try to see the your input file, you will see that there is groups present in your input file, right? Group 1 and group 2, right? So this particular clustering model don't take this groups into consideration. So the segregation of this particular under this particular PCA will be not dependent on groups, right? So this particular PC1 and PC2, I will tell what it is. So go to the score plot, click to the score plot. So once you are clicking to the score plots, you are getting something like this, right? So the blue line, which is at the down, is something which tells you the PC principal components percentage. For example, it is starting with 21.3%. That means the 21.3% of features that is present in your data is having maximum variance in terms of is having the maximum variance. But over here, it is not considering the groups. Then the next cohort is 12.9%. Next is, I think, 8.4%, 6.8%. So once the PC components are going down, the number of the percentage of features are also going down, and make sure this particular 21.3% and 12.9% are not having equal is not having the same features. And obviously, once whatever components you are taking, the components will never give you a cumulative value of 100%. Reason is that the 100% of the data cannot differentiate or cannot segregate your samples or segregate your groups. So as you can see at the top, the green color is showing you the cumulative PCs, like a cumulative score of the components, right? So now I will request you guys to click on the 2D score plots, right? So once you are clicking on the 2D score plots, what you are getting is that the two clusters you are getting based on your group 1 and group 2. But as I told that this particular clustering algorithm don't consider the groups. So that's why you can see, right, that the samples are the clusters, two groups clusters are not much segregated, rather it is overlaid with one another. That means it is telling, right, it is giving an idea that there is around 21.3% and 12.9% on the base of PC1 and PC2. The variations are there, but this variation might not be on the basis of the groups that we have chosen. This could be a biological variation or could be a subgroups that is present in the particular this particular clusters. So, and we also might know this particular PC, the components which are giving, 21.3%, 12.9% is actually reducing the dimensionality of our data, right? And PCA works very good with large data sets, like a kind of omics data set, where the dimension of the data is pretty huge, and it helps to reduce the dimension. And people, use particular PCA to, understand and, select features, even for selecting features on the basis of the eigenvalues, right? So the next part, if you go to the loading plot, the loading plot is something which is a kind of 2D plot on the basis of the proteins, that is the features that is present. For our case, it is gene. right? So the 2D plot, whatever you show, score plots which you have seen is on the basis of the samples, right? And this is on the basis of the proteins or genes, whatever is present as a feature, right? So the 3D, then next click in the synchronized 3D plots. This is something which visually represent the 3 dimension, that is PC1, PC2, and PC3. It's adding another layer of the principal component to show in how the samples are, looking when another component or a 3 dimensional component is added into this particular thing, right? So now I will request you that please come out of the PCA and again go back to the menu. You might see that particular thing is in the left hand side also, and I will request you that click on the PLSDA, or the one which is telling us partial least square discriminant analysis, right? So once you have clicked in the PLSDA, I will request you to click into the 2D score plots, right? So once you have clicked in the 2D score plot, you are getting two different clusters, right? So what is the difference between this PCA and PLSDA? So PCA and PLSDA, one PCA is unsupervised and PLSDA is completely supervised. That means it is taking the groups into consideration. PLSDA works on the principle of linear classification. So what does this particular linear classification does is that it takes the groups into consideration, and it's try to find the features on the basis of the covariance of the features and the protein samples and the groups. So on the basis of the samples and the group, it finds the covariance, and on the basis of the covariance, it's try to segregate the samples and the cluster on the basis of the groups from one another. So that is why you are seeing such a good clustering segregation clusters in PLSDA. Right. So this is the difference between particular PC and PLSDA. I will request you all to read more about PC and PLSDA, because this is something which cannot be, discussed in a 1 hour session, right? And it will give you a lot of information. And I am telling you guys that this particular clustering segregation techniques and this classification techniques can help you to understand about the sample lot more, interpret about the samples much more than what you are seeing in the Excel sheets, right? So another thing is that for any of this Metaboanalyst, figures, if there's the option of kind of paint option is there, and I will if you are clicking this particular option, you will find a high resolution image where format is PNG and resolution is 300 dpi, that is dot per inches, and then the size and everything is available. So you can download all these figures in a high resolution images of 300 dpi, and directly you can use this particular images for your manuscripts, right? So yeah, so I think these two techniques were these two analyses were very important, and I will request that you should go through more study materials on this part. And the next part is that the clustering, which comes on the clustering analysis, is the heat map. I will request that please click on the heat map part, and I will try to give you some idea about the heat maps. So I am focusing mostly on those aspects which you cannot easily do in your Excel or spreadsheets, right? For example, the t-test, the fold change calculation you can easily do in the Excel, but you cannot generate heat maps, you cannot generate PCA and all these things so easily in your Excel. So for this kind of purposes, this particular software can help you to give the idea of the data and give and visualize your data at the same time, right? So hierarchical clustering heat maps is something that will give you an idea of the expressional difference of your features in terms of samples, right? So over here, the two important part is the distance measures and the clustering algorithm. So over here, the distance measure over here, the mostly preferred is Euclidean distance, and we all know about that particular Euclidean distance, that if you are having one point which coordinates are x1 and y1, another point whose coordinates are x2 and y2, the distance will be measured between the x1 y1 and x2 y2 is the Euclidean distance, right? And so there are many other distance calculation techniques are there. For example, Manhattan distance are there, which can also be taken into account. But I will suggest that you should go with for the particular distance measurement, you should go with Euclidean distance for this particular heat map. Then the second is the clustering algorithm of the word clustering algorithm. So there are multiple clustering algorithms are present in the drop down menu, as you can see, but word is something which works on the basis of the agglomerative hierarchical clustering. So agglomerative hierarchical clustering is a very strong, powerful clustering algorithm that mostly work when you are having multiple samples or data points. Let's take an example of data points, and the clustering start based on each data points. So for example, there are another clustering technique which is known as divisive clustering technique, which will start clustering as a it will take all the data points into account, and then it will start the clustering. But over here, the agglomerative clustering works in the bottom up. Right? That means it will select each samples into account and then start small cluster. That means which samples on the basis of the features are coming close to each other, and on the basis of that it will give the clusters of the sample. So this is very important, and I will request that please go through read more about the agglomerative clusterings and divisive clustering, and which will give you an understanding that why word clustering technique is very important, and we should use this part. Then I think there is some color contrast which you can check the drop down menu and you can use. The data source is normalized data we are using. Standardization is absolute scale features. That means from, for example, from -4 to +4, -2 to +2, where the data being scaled over here. Then the view mode is the overview mode. Even there's some detailed view mode is also there, which you can try. Then particular we are using either t-test or ANOVA, and over here, as you can see, that it is showing for the particular all the features, how it is looking in the particular heat maps, and you can see that all the features are not able to segregate or segregate the cluster, that is group 1 and group 2 into two different parts. So that is the same thing we see in the PCA, right? In the PCA also we found that the features, even the 21.3% of the features couldn't 21.3% in PC1 and 12.9% in PC2 could not segregate the samples from one another, and could not make two different clusters, group 1 and group 2 separately. So even we can see the same thing over here. But so that is the reason I will request that you go and use top 25 over there. So this top 25 and click into the submit button, you will find that how the top 25 features are actually looking. So I must say that the top 25 features of when we are taking the top 25 features, and these top 25 features are selecting based on the statistical test, and you can see that this particular top 25 features can actually segregate, except one sample, which you can see this one can segregate into these two groups, that is group 1 and group 2. You can again reduce the features to maybe top 15, top 20, and you can see whether the segregation is more better or not, right? So it will not only giving you the visualization of the expression of your data, but at the same time it is giving you the top 25 proteins which can actually differentiate or can actually segregate your group 1 and group 2 on the basis of this protein. So you can even take these proteins forward and can check that is there any relevance or biological relevance that this particular proteins are separating these two groups, or for your case it can be control versus test, right? So once this part is done, I will just show you some small couple of things, like for example fold change analysis. So this particular thing for the fold change, you can put a threshold of 2, but make sure that in the y-axis it is locked to fold change, which is 1, and it is giving you that how many are upregulated and how many are downregulated, but make sure it is not giving on the basis of the statistical test. This is only fold change, not on the basis of the statistical test, right? So I will request you all to come back and click the volcano plot. So this is another important visualization plot which which you can use in the manuscript to give an understanding of the features in terms of statistical test. I mean significance and also fold change. So the particular plot will give understanding of both. And as you can see the parameters, you can put the fold change as 2, comparison 1/2 means whether you want to calculate the fold change based on uh, 2 versus 1 or 1 versus 2. For your case, it can be control versus test or control versus disease, disease versus control. Anything is fine. And you can put a p-value threshold of 0.05. And obviously, in our case, if you see the samples, you will understand that we have a unequal group variance, right? So as you can see, this particular volcano plot is not looking very great. Reason is that I have given you a subset of a big data, like it was having almost 6,000 proteins, and from that I have given only a list of 1,000, 2,000 proteins, right? So that is why it is not looking very good, but actually you should get a V kind of volcano plot where the red over there, as you can see over there, these are upregulated and these are downregulated based on the log2 fold change, and the cutoff of log2 fold change you have put 1, the log fold change you have put 2, right? And over here, the log 10p is the p-value significance code. That means it has been converted to log 10p, whatever the p-values were there, right? But please make sure that over here, even if you are doing any statistical test, you should be very clear about the which statistical test you should use when you are normalizing your data and your distribution curve is showing normalized, or your distribution curve is showing normally distributed. Go for statistical tests which are parametric. For example, student t test, right? But when your data is not normalized, or you haven't done the normalization of the data in many proteomic analysis, we don't prefer to do normalization. We want to keep whatever the data we are getting. And during those kind of cases, please choose non-parametric test. That means the Man-Whitney test, Wilcoxon's rank test, this kind of test you choose, right? So over here in volcano plot, it gives a very good idea of, if you click in any of this particular feature, as you can see, if I'm clicking over here, it will give the particular protein in terms of box plot, right? And it will also give, and the black dots over here are nothing but the samples. So it is a kind of a swarm plots followed by box plot, and how this particular plot is looking, how this particular protein is looking in terms of 2 cohort it is giving. And this yellow color dot in between is the central tendency, and I suppose it's median, and on the base of that. But if you know some programming language like R and Python, I will request that try to generate these box plots or violin plots in Python. It will give more resolution, and it will give more it will give more clarification of the data, right? So now the next part is that there are many parameters, or there are many analysis there that you can do. And I will request you guys to check each parameters. It will give you an understanding of the statistical test, statistical analysis lot more. And try to Google multiple parameters. You will understand more about those parameters, right? But once you are done with your analysis, just come down and click into the download. So once you are clicking into the download, it will take you to a result download page, and you can click into the generate report to generate a PDF report of this whole analysis. And you can also click into the download.zip to get all the informations and all the files, all the figures that you have generated during the process. Another thing which I missed during the analysis is that during the whole course of time, I will request you guys that who have some idea in the R and R programming language, that please check this R command history, which is always available in the right hand side of your Metaboanalyst. And this particular R command history will help you to understand that what commands are actually there, and how these plots are getting generated, right? And who don't know much about this programming language, I will still ask those people to just try to see this particular codes. Then you will get familiar, and when you will start coding, it will be easier for you to grab, right? So I will try to give you a schematic outflow. What we talked is like, for example, you have got the raw files, and as after Professor Sanjeeva's session, you understood that getting raw files is not very much difficult right now, right? You can get it from Proteome Exchange if you don't have your own data. And lot of data are available, and you can really download those data and reanalyze those data. So you can download this particular raw files, then you can go to MaxQuant, and you are very much right now comfortable with MaxQuant, right? And you can analyze those particular raw files into the MaxQuant. After MaxQuant, you are getting the protein identification. That means the out from the output file, right? So this is something, the correlation matrix thing, which you can even do in over here in the Metaboanalysis. So this is something you want when you want to see correlation between the samples, right? And this is very important to understand that if any sample is having a bad correlation, or the correlation coefficient of the sample is pretty low, then you can check that particular samples, and you can remove that particular sample before you take all the samples for the statistical analysis, because you should not remove samples after doing the statistical analysis, which is not ethically correct, right? So now after correlation matrix, you can go for missing value imputation, followed by data normalization and statistical test, which includes the statistical t-tests and the fold change and all these things. And finally, you can visualize this particular differentially expressed protein in terms of heat map and volcanoplot, right? And once you have done this, you have got a handful of proteins from a list of 2000, finally a list of 20 or 10 proteins, and you want to take this particular small list of protein into the targeted validation, or in the targeted proteomics, where you can use about the SRM atlas, which I think Sai Charan and Medha will talk about this session shortly, right? So I have given some logo of this softwares which we heavily use in our lab, and you guys can also check this softwares. And I think most of the softwares, except few, you will learn today, like Professor Sanjeeva told about Human Protein Atlas, and you will learn about Skyline. I told about Metaboanalyst, right? So now the thing is that the data analysis is not only restrict to Metaboanalyst, and once you are getting a very good idea in Metaboanalyst, I will suggest that come out of that particular software and start exploring more in R and Python. So this particular coding language platforms will give you and an idea that what we can do with the data, and what more we can do, what different kind of plots we can do. And for example, Metaboanalyst give the distribution curve, which gives the total distribution curve of the whole data, right? But if you are like generating a plot in Python, you can see how the each samples are looking, how the distribution of each samples are looking, how the distribution of group 1 and group 2s are looking. So that will give you an idea whether your samples need scaling or not. So these are very important parts, I think, which you can obviously see once you are done with the Metaboanalyst, and you can try in Python and R, right? And there is another software which is Prodigy, which is a very good software for proteomics. It's a R software which can be used, which can be accessed through some very preliminary commands in R, and it has been developed by the Broad Institute, right? You can use that one also. This is also a pretty good software. And but as we most of you are starting analysis for the first time, we felt that we should start with a very user-friendly software, which is Metaboanalyst, right? So I will request all of you that keep exploring, and please get back to us and please ask us questions through email ID, and I will be happy to reply to your mails. Okay, thanks a lot.